The roots of education are bitter, but the fruit is sweet. A quote by Aristotle. Welcome to AHS Guide, your companion for mastering the 5 subjects of first year Allied Health Sciences as per Rajiv Gandhi University. We make complex concepts simple, clear, and exam ready, help you learn smarter, score better, and build a strong healthcare foundation. The topic for today is from Microbiology, the sterilization and disinfection. In today's class, we'll begin our study of sterilization and disinfection, a cornerstone of microbiology and infection control. So it's an important topic because sterilization and disinfection, without this process, the study of microbiology is very difficult. It plays a very important role while studying the microbes. It is essential to keep the sterilization and disinfection as an important aspect. Our focus will be on the introduction and the definition of various terms used in sterilization and disinfection. The various methods of sterilization will be starting with the sunlight sterilization and the heat sterilization. Sunlight is a natural but a limited method, and heat sterilization, we have two different methods, which is dry heat method and moist method. In today's video, we'll be covering about dry heat method, which includes the hot air oven, flaming, incineration, and other applications. This is the first part of our sterilization series. In the next video, we'll continue with other methods such as moist heat, filtration, radiation, chemical agents, and the various other topics that is used for disinfection and sterilization. By the end of this session, you will have a clear understanding of the fundamental principles and the dry heat methods that are used and important and vital in laboratory and clinical practices. So the contents that will be covered in the sterilization series are the introduction, which involves certain definitions related to the disinfection and sterilization, the methods of sterilization, which is in the form of physical method or chemical method that is being used in clinical and laboratory applications. Then we'll be studying about the testing of disinfectants as well. So the introductory part, microorganisms are ubiquitous and can cause contamination or infection. The aim of sterilization and disinfection is to remove, destroy, or inhibit the microorganisms from materials, surfaces, or tissues. So microorganism is found everywhere. You can see in the image there is a presence of good and bad bacteria or microorganisms that is present in our surroundings. It is on everything. It is on the materials, it is on the surfaces, it is on the tissues of the human body. It is present on every possible surface that is around you. So these can cause contamination or infection to the human body. So what is the aim of sterilization or disinfection? It is to either remove the microorganisms completely, or to destroy them completely, or to inhibit their action against the tissues or surfaces that the microorganism is residing on. The definitions of few such terms are given here. Sterilization. This is a process of making an article, surface, or medium free from all microorganisms, which includes both vegetative form or the spore form, and this is achieved by physical or chemical methods. Now, sterilization is a process where you completely remove the microorganisms from certain article, certain implants or surgical equipments that you use, or for any equipments that are being used for human that is going inside a human body or blood tissues. So sterilization is essential to completely remove the microorganisms from that material or surface or an article or a medium. Now, how is sterilization done? It is done by physical and chemical methods. Physical method is by doing a mechanical work or producing certain temperature or a certain temperature of heat to the organism, so that the organism as well as the spores that is being produced during reproductive stage of the microorganism, it is removed. Chemical methods are usage of certain chemical disinfectants that are going to kill the microorganisms. Now here, the vegetative forms are the ones like the microorganism, the cells that you see. The small dotted structures that you see are the spores that here. So spores is a unfavorable condition. These spores are going to develop into microorganism when a favorable condition is present. So it is essential to remove both the vegetative form and the spore forms here. So note that sterilization involves removal of spores as well, along with the vegetative microorganism, which is a living microorganism. So moving to the next definition, which is disinfection. This is the process of destroying the pathogens or the microorganisms which are capable of producing infection. This disinfection does not require removal or complete killing the spores. So the disinfection is going to reduce the microorganism to a safe and a non-harmful level. So we have seen disinfection happening in our COVID times. So during Corona, the process of disinfection was taken as a vigilant process to remove the microorganism that was causing the coronavirus or any other microorganisms that's going to aggravate the situation of corona as well. So it was essential to disinfect the infected regions where the COVID infected patients were there. So this process of disinfection, it involves the removal of the pathogens that is causing diseases and the ones which are going to cause infection as well. So these are completely removed. So here in disinfection, there is not necessarily that the disinfection must involve the spores as well. So it does not require the spore removal as well. But here the vegetative form of pathogens or the organisms can be removed. So this disinfection is going to be a safe and a non-harmful level. It is not going to cause any harm to the microorganism, but it is going to reduce the number of microorganisms. So keeping the microorganisms, the good microorganisms is also essential in the surroundings because they are going to be helpful for the biosystem to be balanced. So it is essential to keep the microorganisms in spore formation, not exactly in the vegetative form. So disinfection is used in destroying the vegetative forms. So there is a difference between disinfection and sterilization, and that is essential to know. Moving to the third type of definition, which is the antiseptics. So these are the chemical disinfectants that was widely used during COVID times. The sanitizers that you guys applied every day after touching any surfaces because of the COVID. Now here, the chemical disinfectants that can be safely applied in to the living tissues. So these are not consumed inside but can be applied on the surface of the living tissues. So here it is used to prevent the infection by inhibiting the microbial growth. So when the microorganisms on is residing on the living tissues, that is on your hands or on your legs or any other region, you can use the chemical disinfectants such as alcohol or chlorhexidine, povidone, iodine. So these are the different forms of chemical disinfectants that were used to remove the microorganisms and also inhibit the growth of microorganisms on the living tissues. Moving to asepsis, this is a technique used to prevent the entry of microorganisms into an uninfected tissues. This procedure is used in surgical and clinical practices, and these are carried out in a germ-free environment. Asepsis is the process of A meaning removing, sepsis meaning the bacterial infection. So to remove the bacterial infection, this procedure is adopted. So here the technique is used in such a way that it is going to prevent the entry of microorganisms into an uninfected tissue. So that how if a region, say you have a pus on the hand, so the pus that is involved in that region is going to be removed in such a way that the healthy tissues around the pus is not affected. So that is the process of asepsis. So your surgical or clinical procedure that involves that is done in a germ-free environment. So here you can see the germ-free environment. There is no microorganisms. These are all sterilized. So these are free of all microorganism, either in vegetative or in the spore formation, but sometimes the patient might have a infection inside a certain region where it is infected with the bacteria. So you are going to remove the infected region by not touching the uninfected part. So that is the role of asepsis. The use of sterilization and disinfection. Medical and surgical fields, sterilization is essential and is performed for the instruments, the dressings, the syringes and catheters that is used in the surgical field. Diagnostic procedure are safe handling of samples and instruments. So sterilization, disinfection can be done in the diagnostic procedures where you need to handle the samples and instruments. Now, microbiology laboratories, certain media require sterilization like glasswares and reagents. So these are required to grow the certain type of single type of microorganisms. Then sterilization is essential to avoid any the surface the microorganism that is in air to enter into the media. So sterilization is essential in the microbiology laboratory. Moving to the fourth type of use, where food and drug industries also ensure that the safety of products by preventing the contamination. So once the food or in the drug industry, the food processing is happening, microorganisms can contaminate or infect the food products or the drug products. So it is essential to prevent the contamination either by disinfection. Now, research settings also apply the sterilization and disinfection in their routine practices. It is used to maintain an aseptic condition in culture and experimental work. So you can observe that there are glass shields in between the sterile field or the microbial lab, and it is very vigilant that the sepsis is not happening. So the bacterial infection can come from outside to inside or from inside to outside of the laboratories. So it is essential to keep the area in aseptic condition. There are going to be ACs that are going to there are going to be air conditioning systems that are going to circulate the infected air outside, and there are strict vigilance about the PPE kits, that is personal protective kits in the research settings. So it is kept very vigilant and very important for the aseptic condition in order to carry out the culture and experimental work. Because of the sepsis, the infection might spread into the community, like the COVID happened. There was an accidental spread of the COVID virus, the coronavirus, and it led to a very devastating situation for the whole world, right? So it is essential to keep the sterilization and disinfection process in check all the time. Now, the methods of sterilization that can be adopted in clinical and a laboratory setting are the physical and the chemical methods. Now, today we'll be covering the physical methods and the sunlight, heat, and dry heat alone. In the next videos, we'll be learning about the moist heat, ozone, filtration, and radiation. The chemical methods will be covered in other video, and the last topic involving tests for disinfection will be the fourth video. So in this video, we'll be studying about the physical methods. Now first we'll see the methods that are being used in sterilization as an overview. The physical methods here involves sunlight, heat, ozone, filtration, and radiation. So sunlight is a natural method of sterilizing material or a surface. So how do you do it? You are going to expose the material or an article or a surface that has to be sterilized into the open sunlight. So this sunlight is going to kill the microorganisms which are present on the surface. So it's a natural form and it is very limited. So sunlight is not going to be produced for a whole 24 hours. It's just going to be 12 hours a day. So and the effective part of sunlight is going to be around 3 to 4 hours, just 3 to 4 hours. So this is a natural form with a disadvantage of sterilization. Disadvantage during due to the time constraints of the sunlight. Now heat is another method that can be used in the laboratories, like the Bunsen burner that we have been seeing from our 6th to 7th standards. These Bunsen burners can be used for dry heating. Sometimes there are certain machines that are being built for sterilizing microbial media, culture medias, or the surgical equipments that are used, and various other medias that has implants or any other media that has to be sterilized is going to be going through this heat sterilization physical method. And the ozone is another method where oxygen, the nascent oxygen that we have is going to be used for removing of the microorganisms. So ozone plays an important role for killing the aerobic and anaerobic microorganisms by producing the toxic form of O3, which is going to kill the microorganisms. Now, filtration is another method where bacterial filters are used. It is a type of sieve kind of a thing which filters out the bacteria. But the disadvantage here in filtration can be the viruses are very small compared to the bacteria. So viruses cannot be filtered by this method. Radiation is another method that is used to kill the microorganisms. We'll be dealing with it in another video. Now, chemical method here involves the various types of disinfectants, which includes alcohol, aldehydes, phenols, halogens, oxidizing agents, salts, surface active agents, dyes, vapor phase disinfectants. So these are the different chemicals that are used to sterilize. The physical method of sterilization, the first type of physical method is sunlight. It has a germicidal effect due to the ultraviolet rays, a natural method of sterilization, and it is useful in water tanks and rivers and lakes. So sunlight was in olden days being used to dry up all the materials that was being used in homes. So they used to dry it in the sun, keeping it outside in the sun. So it was a method of sterilizing the articles back in those olden days, because they had a germicidal effect. That is, they can remove germs, can be killed by this ultraviolet rays that is being produced by the sunlight. And this is a natural method, obviously, and it is useful in water. So tanks, rivers and lakes are said to be sterile or free of any harmful microorganisms because of the sunlight. Now, heat is a most reliable and commonly used method of sterilization. So at certain temperature, the microorganisms can be killed. So the principle that is being used for a dry heat and a moist heat sterilization is being here. In dry heat, it kills by denaturation of the protein, oxidative damage, toxic effects of electrolytes, and possible DNA damage. So what does this tell? Now, kills by denaturation of protein. In biochemistry, we have learned about the proteins, and under proteins, once the breakdown of proteins because of the breakdown of the structure of the quaternary structure of the protein, the proteins cannot function efficiently. So if the structure of the protein is destroyed because of the high temperature, the bacteria cannot work, or the microorganism cannot work without the protein's effect. So once the protein effect is not working, then the microorganism is going to be dead. Now, oxidative damage is by the oxidizing effect, that is, either the electron is removed from the any molecule that is present inside the microorganisms, then the removal of electron essential for the structure of a microorganism can also cause damage to the cell. Now, toxic effects of electrolytes, that is the osmosis process that is happening, there might be excessive of outflow of electrolytes or inflow of electrolytes, which can lead to death of the microorganisms. There are possible DNA damage also because of the high temperature. Now, moist heat is another method of sterilization under heat. Here it kills by denaturation and the coagulation of protein. So due to the excess of temperature, the protein structure is broken down, and the coagulation of the protein, that is the lump formation of protein inside the microorganisms, can kill the microorganisms. The factors that are influencing in the sterilization by heat is the nature of heat. It can be dry or moist. Then the temperature and the duration, which is inversely proportional. So whenever the temperature is high, the less duration is required for sterilization. If the temperature is low, more amount of time is required to sterilize. So that is inversely proportional. Now, spores killed by moist heat is, as you can see, at 121 degrees Celsius, which is very high temperature. It requires only 15 minutes to kill the spores, the unfavorable condition of microorganisms to grow. That is the state of a spore. Now, vegetative bacteria, fungi, viruses can be killed at 65 degrees Celsius for 30 minutes. So, at a 65 degrees Celsius, it will require around 30 minutes to destroy the bacteria and fungi. So, at very low temperature only the vegetative forms will die, but the spores require very high temperature. Now, types of microorganisms, spores are very much more resistant compared to the vegetative forms. As you can see, the temperature that is required for vegetative is less, but for spores it requires 121 degrees Celsius. Now, the types of material that is present on the microorganism will also determine the amount of heat that is required to sterilize. So, the organic matter such as sugars, fats, starch, proteins are going to protect the microorganisms, which require longer sterilization. So some microorganisms can have a protective layer of the organic matter, which is going to prevent its easier desiccation or easier death. So these protective layers need to be broken down. So in order to break them down, it requires a longer sterilization. Now moving to the very first type of sterilization under heat, which is dry heat sterilization. The methods that are adopted for a dry heat sterilization are red heat, flaming, incineration, and hot air oven. Yeah, that is what we'll be covering. So there are 4 different types of dry heat sterilization: red heat, flaming, incineration, and finally the hot air oven. Now red heat, here the inoculating wires or loops, the tips of forceps, needles held in Bunsen burner till red hot. It is very simple. Here the sterilization is performed by placing the tips of the or any instrument that you are using for handling the microorganism before you touch into the broth or the specimen samples that you have to get a culture of. You are going to place the inoculating wire. It is a wire that is used to dip into the human specimens that you take from the laboratories and do the culture in the laboratory. So here you use the inoculating wires or loops or the tips of forceps or the needles and place it on the Bunsen burner. You'll observe a red hot color on the tips of these materials. Once it is red hot, you can take it from the flame and it is sterile. So that is the method of red heat. Now moving to the flaming, here you are simply going to move the substance that is being move the surface or the substance that you are using for sterilization onto the Bunsen burner. You're not going to make it red hot or break it. You're not going to give high temperature to it. Here you're just going to move or pass over the flame. So which all are going to be considered for flaming are glass lights, because you can't burn them red hot because they'll break. So glass slides, scalpels are the surgical knife that is being used. Culture tube mouths, so whenever a culture tube is used. Moving to the next type of dry heat sterilization, which is incineration. This process involves the infected material is going to be burned into ashes. It is usually done in incinerators and used for soil dressing, animal carcasses, bedding, pathological waste. So incineration is the process of completely removing the biological material that is being used on the patient or the animal carcasses. So animal carcasses are the ones that are remains of the surgical procedures. That is, the infected materials from the animals can be burnt out. So here the complete heating until the material is completely burnt into ashes. So this is a method of completely removing the pathological waste in order to avoid the infection to breed on these pathological waste. So here is an example of how a red hot sterilization done. Flaming, that is test tube is passed over the heat, and then we have the incinerator wherein the pathological waste can be completely burnt into ashes. These are the three different types of dry heat sterilization. Moving to the fourth type of dry heat sterilization, which is hot air oven. This is commonly found in the microbiological labs, wherein it is widely used for dry heat method, and it is electrically heated. A fan for uniform distribution is placed inside the hot air oven. So as the name says, hot air and oven, the air is heated up and that air circulates inside the chamber and creates a heat sterilization effect. Here it is controlled by a thermostat. So a thermostat is the one which can control the temperature at a certain period of time. The same temperature is kept constant, so that is controlled by a thermostat. And here the temperature is usually around 160 degrees Celsius for 2 hours. That is a standard temperature and is maintained for most of the articles that are being sterilized through dry heat sterilization or hot air oven. Here the alternatives can be around 170 degrees Celsius for an hour or 180 degrees Celsius for 30 minutes. So it can depend upon the amount of material that is being placed or the amount of sterilization the culture media or any other material requires. So based upon the requirement, you can change the temperature and the time, that duration of sterilization. So standard one says 160 degrees Celsius for 2 hours. So this you can mention in your answers, and alternatively you can mention 170 degrees Celsius, but don't get confused. The standard method of representing the temperature and time for hot air oven is 160 degrees Celsius for 2 hours. Now, which all materials are being sterilized using hot air oven? One is glassware, which will involve syringes, petri dishes, flask, pipettes, and test tube that are used in the laboratories. Surgical instruments are also sterilized by this method. That is, the scalpel, scissors, and forceps are sterilized by this method. 2 hours before the surgery or 1 hour before the surgery, the instruments can be kept inside and sterilized. Now, chemicals that are used for the culture media of bacteria or a microbiological culture media. Here the chemicals are also sterilized if in case it is done in a anaerobic chamber or something like that. Now, what are the different chemicals that are sterilized by this method? Method is liquid paraffin, fats and powders, sulfonamides are also sterilized by this method. Now here is a picture of how a hot air oven looks like and how the instruments are placed inside. So there is a temperature that you can set, and the duration of the temperature can also be set, and there are certain controls also present in order to control a hot air oven. So these are electrically operated and they consist of a fan inside for the uniform distribution of the hot air in order to sterilize through dry heat. So there is no moisture or no water added to this. The air mixture is only heated and sterilized. You can see the surgical equipments are also kept inside a hot air oven for sterilization. So the precautions that you have to note while doing a dry heat sterilization are, it should not be overloaded, because overloading can lead to infrequent sterilization by the air, or there is no proper sterilization, and overloading can also lead to breaking of the instruments inside. So it is essential to keep a proper load of instruments that has to be sterilized. Secondly, arrange the materials for free air circulation. Since the air is being circulated through the fans, the arrangement should be in such a way that the air circulation is easy. The third type of precautions is material should be completely dry and there should not be any water molecule present inside. If it is present, a water molecule is present, then the sterilization is not possible and the microbes can be there in the water, which can lead to improper sterilization. The fourth method is a test tube or flask should have a cotton plug in order to avoid the flow of hot air into them and the cracking of the test tubes of flasks. Petri dishes or pipettes are also should be wrapped in paper in order to avoid direct exposure to the dry air or the hot air. Now avoid rubber because rubber can be melted or it can cause any fire accidents inside the hot air oven, which is fatal. It can lead to bigger blasts. So rubber should be avoided and all the flammable should be avoided. The seventh precaution that you should follow is the oven should be cooled for 2 hours before opening. So sudden open, once the sterilization is over, do not open the door of the hot air oven directly. It can lead to cracking of the glasses or the glassware that is being kept for sterilization. So it is essential to cool the oven for 2 hours, and after 2 hours, so 2 hours of sterilization, 2 hours of cooling, and then you can open the hot air oven and retrieve the equipments. Sterilization control. So this is the method that is used to check whether the equipments inside are sterile or not. So these sterilization controls are being used as an indicators for the sterilization. The first method is a biological indicator, which is Bacillus subtilis spores. So these are a bacterial spores that are being present on the tape. It is like a tape kind of a structure wherein you can stick the tapes on the equipments that you are sterilizing. So once these Bacillus subtilis spores are exposed to the dry heat sterilization, they are going to show the indication as a black color strips. So this can prove that the equipments inside are sterilized. Now thermocouples is a monitor temperature. So these are present in the machine itself or the hot air oven itself, which determine the amount of temperature that is being delivered inside the chamber. So these are going to monitor the temperature. Now Brown's tube is again a indicator which has a red spot. It turns into green after 2 hours, that is after sterilization at 160 degrees Celsius. So here is an image of the different indicators that are being used inside the hot air oven. So firstly is the strips that are used for checking the sterilization. Here these strips are going to determine the spores that are being exposed to sterilization and the death of the spores. So they have mentioned dry and these are going to be green in color. And then we have the thermocouples. These are going to be placed inside the tubes or the equipments that are kept inside the hot air oven. So these are going to keep the temperature at a constant level. So these are like a temperature monitors. Now we have the Brown's tubes which are used for checking the sterilization level inside the hot air oven. So these are directly placed into the hot air oven and checked for the change of color. So if the red color is changed to green, it is indicated that the sterilization has happened. If the color does not change or any other color is appearing, then the sterilization has not happened. So that is the indicator of hot air oven, dry heat sterilization in a hot air oven. So now we have covered the various aspects of sterilization and disinfection and its role in microbiology as well, and various other clinical practices. So it is essential to learn about the sterilization and disinfection in order to understand how the microbial lab without any infection, or the precautions that the microbial labs are taking in order to prevent the exposure of the infection or to the general public, or contracting the infection by a professional healthcare worker. So we have covered the sterilization definitions of sterilization, disinfection, asepsis, and asepsis and various other terms. We have also covered the methods of sterilization. The first part of methods of sterilization, which is physical method, we have already covered the sunlight, heat involving dry heat sterilization. So in the next video we'll be doing about the moist heat sterilization and rest of the topic. Till then, keep watching AHS Guide.